So let's get back into our work, and we'll start with blood. Small introduction to what blood is. Well, it's one of your fluid compartments. So do you remember in the body fluid compartment video? If you haven't, I recommend you go back and have a look at that. Your total body water, if you look at it, is divided into intracellular fluid and extracellular fluid. Do you remember that? And the ECF was divided into plasma and interstitial fluid. Well, I had this diagram drawn before where we had the blood vessels in the vascular compartment and various cells in the diagram. So this diagram represented the distribution and the composition of the body, the distribution of fluids in the body. And here I said, you take your blood vessels over here and you put them in one compartment, and outside here we have these different cells. This could be a liver cell, a kidney cell, a skin cell, whatever. All of these are the different cells outside the compartment. Well, blood it belongs to the vascular compartment. This is the vascular compartment, or you could say the vascular tree, because sometimes you see this diagram where there's the heart and you see that blood goes from the right ventricle to the lungs, and there's this particular diagram, something like this, where there's the lungs, and from the lungs it goes over here, and then from here to the various organs. You get these capillary tufts of capillaries through the various organs in the body, the visceral organs and the different types of tissues over here. So there's this kind of a circuit, and it resembles a tree if you look at it, and the blood goes back to the right atrium over here. It's a rough diagram, please excuse it, but understand that this kind of looks like a tree with branches and little branches over here. So this fluid, this red fluid which passes through the vascular tree, your arteries, your veins, your capillaries, and through the chambers of your heart, this is blood, this red color fluid over here. And we're going to go into detail as to what it is, how it works, what its functions are, and how it carries out these different functions. You will be studying a lot of this in pathology also, and right now this topic is more geared towards understanding the physiology, which is my main forte, but we will have a little overlap in what in various pathological topics also. So let's get into it. So here, blood, if you look at it, it's a very interesting fluid compartment, because if you look at it, it's got intracellular fluid and it's got extracellular fluid in the form of plasma, both in one compartment. So it's interesting that you have both of them in one sort of a compartment over here. These various body fluids in one single compartment. This is one very interesting point to remember. Okay, now the blood vessels are lined by endothelium over here, okay, and fluid will pass through the endothelium and sometimes between the cells, because not all the blood vessels endothelium are next to each other. There's a little space sometimes between them for fluids to ooze in and out of. Okay, anyway, so let's start. What are the functions of blood? So if we look at it, our body has around 5 liters of blood. Right, 5 liters of blood, that's the average amount over there. And if we look at it, it is going to be this one, interesting to know that there are more functions than just the oxygen and CO2 transport that takes place over here, meaning gaseous transport over here. So your lungs oxygenate and give oxygen to the blood, and the blood gets rid of the CO2 in the lungs, and we exhale that CO2 over there. Okay, so we expire that CO2. So there is transport of gas component over there, so transport is very important, of course, and that's the primary function, you could argue, of blood, because if it doesn't transport any of these gases, then you die, because no oxygen means no way for mitochondria to make ATP, and no ATP means no energy, and you are dead. And CO2 is quite toxic in high amounts, So there is a limit. We have a requirement for CO2 in our body, but if we get rid of if we accumulate all the CO2, it's not going to be good for us. Okay, so transport of gases. Then we also transport other things like nutrients, other metabolites, and chemical messengers like your hormones. So ductless glands exist, like for example your pituitary gland. It's located over there in the brain, and what happens is the anterior pituitary has some cells which secrete various hormones like growth hormone or thyroid stimulating hormone, whatever. Okay, so what's going to happen is, for example, this thyroid stimulating hormone, which is secreted from the anterior pituitary, it goes through the blood vessels of the pituitary to the thyroid gland, and that is going to make the signal the thyroid gland to release T3 and T4, your thyroid hormone, which eventually gives a negative feedback to this. But the point is, hormones get transported to the blood. Those hormones are secretions made by ductless glands, meaning no ducts, no storage. It's just going to, the cell makes the hormone, and when it's stimulated, it releases that hormone directly into the blood. Right? Those are your hormones. Then after that, you have other important functions, like your blood has, apart from the red cells which carry out the oxygen and CO2 transport function, you have some white cells. So those white cells are important in your defense, immunity of the body. So to defend yourself from various microbes and infections. Okay, so immunity against infections. Immunity comes from the word immunos, meaning freedom. So you are free of these infections and ailments because of that. Right? And then you have another thing, which is the maintenance of body temperature. Think about it. Whenever you exercise or your body gets hot, what happens to your face? It gets all flushed up. You get all pink in color. Why? Because vasodilation is happening over there. The blood vessels dilate, so especially in the skin, so that they can radiate and come in contact with the external environment more, bring that warm blood out there to the external environment, so you can radiate and even lose some heat by convection and conduction also. So that's why you have the vasodilation taking place. And when it's very cold, your blood vessels constrict. You could see your hands and legs, the skin on your hands and legs become very pale, even on the face, because the blood vessels will vasoconstrict, pushing that warm blood deeper into the body to maintain that core temperature. Okay. Anyway, so then after that, we have another important function, which is blood is an effective buffer. It's a buffer. You got buffer systems over there. You have these proteins in blood we'll be talking about, which have the ability to buffer the pH, maintain the pH of blood, around 7.4, or if you want to say 7.35 to 7.45. So that's how you maintain your pH. Some books say 7.38 to 7.42. Anything works, whatever. So you can choose any of these numbers. Right? And then after that, you have your other functions, which is the maintenance of of your fluid and electrolyte balance. Fluid and electrolyte balance. Okay, and then after that, you have other functions, like it has a mechanism to protect itself from getting lost. For example, hemostasis. You prevent blood loss by clotting, and these hemostatic mechanisms is a very important topic which we'll be talking about also in the very near future. Alright, so it prevents its own loss from the body. It is important in fluid electrolyte balance. It's a good buffer, so it plays a role in acid-base balance. It's good for maintaining a body temperature, immunity, and also it supplies and moves around nutrients, metabolites, and hormones, and oxygen and CO2 transport. Some of these metabolites are waste metabolites that get excreted from organs like the kidneys in the body. So that's your function of blood. Now let's go ahead a little more in detail with the composition of blood. So remember this diagram over here, which we've come back to. Yeah, this is your vascular compartment. So if we take the chamber, this path that blood moves in, this vascular compartment, just draw it as one nice long cylinder, okay, for simplification's sake. So the heart chambers and the vascular tree we've drawn as one chamber over here. Now your blood cells, they will take a certain volume of this chamber. How much volume is that going to be? Actually, we should just make it a little lesser than half. There we go. That is better. So this is your red cells, which we have packed in one corner over here. All right? These are the red cells. On top of the red cells, let's just maintain a small layer of white cells and platelets. Use your imagination. And a thin layer of platelets. So the plasma can be anywhere between 55 to 60% of the volume of your vascular compartment. This is plasma. 55 to 60%. The cells, especially the red cells which we're talking about, are around 40 to 45% the volume of your vascular tree. So this is your packed cell volume, or your hematocrit. So how do you get this? Well, one way you can separate these different elements and see the proportions of your blood and the various compositions of your blood is you take a little test tube, fill it up with blood, and of course this is citrated, anticoagulated blood. You don't want it to clot by itself. And what you're going to do is you're going to put it in a centrifuge and rotate that centrifuge at 3000 RPM, 3000 revolutions per minute. And as you do it for around a half an hour, these heavier elements, they're going to settle at the bottom over here. The cells will settle at the bottom, while the lighter cells, like the plasma, and even the lighter cells come on top, like the platelets over here. But anyway, plasma is even lighter, so that goes on top. That's got something to do with the centripetal and centrifugal forces over there. So you have the centrifuge, which is a machine which rotates very rapidly, and the heavier stuff goes outwards over there. So by doing this, you are able to separate and stratify the different components of blood over here. And you will notice that the red cells take around 40% of the cells to 45%. In females, it is 40% hematocrit value, females. Right? And it is around 45% for males. And you must also be aware of the fact that these cells they have a shape. I'm going to talk about it in more detail. They're biconcave, discoid shaped over there. If you look from the side, they have concave surface on both sides, but when you look from the front, they're like a disc, but with the peripheries a little thicker, like those donut pillows you get for certain people. So these are, think of a donut, but it's not with a hole in the middle. It's more like thin in the middle, but not hollowed out in the middle. So females, so why am I telling you about this? Because when you centrifugate this blood, there's space between these cells. There's some space between them. Can you see I'm drawing it in green over here? So there's still going to be a little plasma, around 2 to 3% of this volume is going to have some plasma in it. You're not exactly going to get pure cells in this hematocrit over here. You're going to get some plasma over there. Okay, just be aware of that. Now, so like I said, females 40%, males 45%. This is how it is. Why? Females menstruate and males don't menstruate, no matter what anyone feels. And at the same time, males they tend to have more testosterone in the body, and this testosterone is supposed to help them in producing proteins. For example, globin, hemoglobin, which is contained in your red cells. There's a heme component and a globin component. The globin is protein, and this protein is made more thanks to the testosterone. So that's why males tend to have a little more blood than females, provided they are given the appropriate diet for their age and their this one composition. So now let's continue. If we look at the composition of blood, some numbers you are supposed to be aware of. So you have to know the count of the different cells. You have red blood cells, or erythrocytes. We call them erythrocytes. These erythrocytes are, various books give different numbers. So if you're a female and a male, there's a difference, like I said. So females, they have around 3 to 5 million, so million, 10 to the power 6 cells per milliliter of blood. For males, it is around 3.5 to 5.5 million, so 10 to the power 6 per ml of blood. This is your RBC count. And these numbers vary in different books, don't worry about it. With your clinical experience and the place you practice, those and the labs that are associated with your practice, the reference values can change, so don't worry about it. If you know these numbers, that's good enough. Definitely, if a female has less than 3 million cells per ml, she is not healthy and she might be anemic. And if a man is having less than 3.5 million cells per ml, definitely he might be anemic. So that one we're sure of over there. And if anybody has more than 6 million cells per ml, that's a lot of cells, and we call that polycythemia vera. So remember, more than 6 million is called polycythemia, 10 to the power 6. This is called polycythemia. We'll just talk a small amount about this, okay? Polycythemia. We'll get to that, don't worry about it. And then you have your white cells, WBCs, white cells, also known as leukocytes. So your total WBCs are around 4,000 to 11,000 per ml for the adult, for both male and female, male and female adult. Kids have even more than this, depending on their age. It can even be up to 16,000, 18,000. So it's different in children. Right now, let's focus on adults. So we have 4,000 to 11,000 per ml. So let's talk about the adults first before we go into the kids, because it's a little different in children. There are some changes which we'll be discussing about later. Now, the white cells or the leukocytes are divided into two types, which are granulocytes and agranulocytes, based on the visible granules from your GIMSA staining. So there are these types of stains which are called your GIMSA stains, GIEMSA, GIMSA. So these GIMSA stains, for example, Leishman's stain is one of them. You might be doing them in your labs. So they stain these different blood samples, and you will find some white cells are there, nucleated cells, which have granular appearance. They look like they have granules in them. So those with the granules are granulocytes. I want you to understand that this is based on GIMSA staining. GIEM, GIMSA staining. So that's what decides if something is granulocyte or agranulocyte. But please understand, every cell has granules. Every cell, whether they're granulocytes or agranulocytes. What do you mean by granules? Little dots over there when you see on the microscope? Yeah, so those are your granulocytes. Well, the thing is, agranulocytes, they have some organelles called lysosomes and peroxisomes. So they also have granules, okay? But they're not easily visible when you do GIMSA staining, but in GIMSA staining, certain cells, these granules are visible, but in GIMSA staining, agranulocytes, you can't see those granules so easily, but technically they all have granules. These granules are different. The granulocytes have different granules with different compositions, and they pick up the stain much better and are much easily visible.